Good morning all, this is Sanjay doing second year MBBS at Madras Medical College. Today we are going to discuss about general mechanism of cell injury. So cell injury usually results from any abnormalities in one or more essential cellular components. So those components include mitochondria, cell membranes, DNA and any problem in machinery of protein synthesis and secretion. Next, let us discuss about each one in detail. So first let us see about mitochondrial damage. So though mitochondria are critical players in all pathways leading to cell injury and cell death because intrinsic, as you all know, the intrinsic pathway of apoptosis occurs inside the mitochondria. But still mitochondria plays a vital role in supplying ATP and hence known as powerhouse of the cell. Thus, uh, thus, in many ways the injury to mitochondria or any action of stimuli on mitochondria will lead to cell injury and cell death. So mitochondria can be damaged by increase in cytosolic calcium concentration, reactive oxygen species and oxygen deprivation. So which makes them sensitive to virtually all types of injurious stimuli including hypoxia and toxins. So there are three major consequences of mitochondrial damage. The first one is ATP depletion. So decreased ATP synthesis and ATP depletion are frequently associated with both hypoxic and chemical or toxic injury. So ATP is produced in two ways. The first pathway is the major pathway in mammalian cells, particularly which occurs in non-dividing cells such as brain and liver cells, is oxidative phosphorylation. Oxidative phosphorylation of adenosine diphosphate in a reaction that results in reduction of oxygen by the mitochondrial electron transport system. Next is the glycolytic pathway which can generate ATP. So due to absence of oxygen using glucose derived either from body fluids or from the hydrolysis of glycogen. So to that we will get glucose and hence ATP is produced somewhat by the glycolytic pathways in the absence of oxygen. Next, the major cause of ATP depletion are hypoxia, ischemia and toxins such as cyanide poisoning. So the mitochondrial damage will result in the formation of high conductance channel. Okay, high conductance channel in the mitochondrial membrane that is called the mitochondrial permeability transition pore. So opening of this channels will lead to loss of mitochondrial membrane potential resulting in failure of oxidative phosphorylation and progressive ATP depletion that leads to necrosis. Next, high energy phosphate in the form of ATP is needed for all synthetic and degradative processes within the cell. So the depletion of ATP of 5 to 10% of normal levels has widespread effects on many critical cellular systems. We will see what happens if there is a depletion of ATP to 5 to 10% of normal levels. The first one is the activity of plasma membrane energy dependent sodium pump. Sodium potassium ATPase is reduced. That is, the activity of the plasma membrane energy dependent sodium ATPase pump is reduced, and hence it will cause sodium to enter and accumulate inside the cells, and potassium concentrations will fall inside the cell. This results in loss of resting membrane potential. So the net solid gain results in osmotically driven water accumulation. So due to this entry of sodium, the result is that due to osmosis the water accumulates inside the cell and leads to cell swelling and endoplasmic reticulum will dilates due to entry of water into the cell. Next, the cellular energy metabolism is altered. So the supply of oxygen to cells is reduced in ischemia or oxidative phosphorylation ceases. This will result in decrease in cellular ATP and increase in adenosine monophosphate. So these changes will stimulate phosphorylase and phosphofructokinase activities, leading to increased rates of glycogenolysis and glycolysis respectively, in order to maintain energy supplies by generating ATP through metabolism of glucose, which is derived from hydrolysis of glycogen. See, glycogen stores are rapidly depleted. That is an important feature of uh, injury to mitochondria. So therefore, that is the indicative of hypoxia. This glycogen stores are rapidly depleted means it is an indication of hypoxia or ischemia, and oxidative phosphorylation decreases. So in order to compensate that, there will be increased rates of glycogenolysis and glycolysis due to phosphorylase and phosphofructokinase activities. Okay, but in anaerobic conditions, glycolysis will result in accumulation of lactic acid and inorganic phosphates from the hydrolysis of phosphate esters. So due to presence of accumulation of lactic acid, the intracellular pH is reduced and it will result in decreased activity of many cytosolic enzymes. So with the prolonged depletion of ATP, the structural disruption of protein synthetic apparatus occurs. If there is no ATP, then automatically the protein synthetic apparatus disruption will occur, and itself will manifest as detachment of ribosomes from the rough endoplasmic reticulum. So that is, the ribosomes present in the rough endoplasmic reticulum will separate, and dissociation of polysomes will take place with the consequent reduction in protein synthesis. So there may also be increased protein misfolding with injurious effects, and also there is irreversible damage to mitochondrial lysosomal membranes and the cell undergoes necrosis. That is important. Due to incomplete oxidative phosphorylation, there is a formation of reactive oxygen species, that is free radicals, which have many dangerous effects, and the leakage of mitochondrial proteins due to channel formation by pro-apoptotic BAX and BAK is the initial step in apoptosis by the intrinsic pathway. So the action of BAX gene and BAK is specific to mitochondrial membranes only and leads to damage of other organelles indirectly. Next, in membrane damage. So first we have seen damage to mitochondrial damage, and in this flow chart, what we discussed is that ATP synthesis decreased, energy dependent function also decreased, and increased reactive oxygen species will cause damage to lipids, proteins, nucleic acids, and ultimately cell injury and necrosis will occur. Due to reactive oxygen species, there will be damage to lysosomal membranes and damage to plasma membrane due to leakage of lysosomal enzymes, and impaired transport functions and leakage of cellular contents will occur. That is, cell membrane gets damaged and leakage of cellular contents and impaired transport functions will occur, causing necrosis. And then due to mutations and radiation effect, there will be a mutation in nucleus and it will result in DNA damage, and therefore it will ultimately leads to apoptosis. So apoptosis results from leakage of mitochondrial proteins, and decreased oxygen supply, toxins, radiation will result in decreased ATP generation, increased production of reactive oxygen species, leading to necrosis. Next, let us discuss about membrane damage. So we will be discussing about membrane damage and damage to DNA and ROS, reactive oxygen species, so that this mechanical injury gets completed. Okay, so let us discuss about membrane damage. Early loss of selective membrane permeability leading ultimately to overt membrane damage is a consistent feature of most forms of cellular injury. That is, early loss of any loss of membrane permeability, selective membrane permeability. Okay, result is the most common symptom of cell injury or a most common form of cell injury. So in ischemic cell, that is in cells whose blood flow is low, the membrane defects may be the result of ATP depletion and calcium mediated activities of phospholipase. So the plasma membrane can be damaged directly by bacterial toxins and viral proteins, and due to always reactive oxygen species, these are oxygen free radicals which will cause injury to cell membranes by lipid peroxidation. Very, very important. The reactive oxygen species damage the cell by causing injury to cell membranes through lipid peroxidation. Next, decreased phospholipid synthesis. So the production of phospholipids in cells is reduced as a result of defective mitochondrial function or hypoxia. So both of which decrease ATP production and thus affect energy dependent biosynthetic part is very important. This three lines is very important. So mitochondrial function or hypoxia will cause a defect in mitochondria, lead to decreased ATP synthesis, and therefore phospholipid synthesis also will get reduced. Okay, next, due to increased phospholipid breakdown. So due to activation of calcium dependent phospholipase by increased cytosolic and mitochondrial calcium levels, phospholipid breakdown leads to accumulation of lipid breakdown products including free fatty acids, acyl carnitine and lysophospholipids, which will have the detergent affected membranes. That is, they will insert, either insert into lipid bilayer of the membrane or exchange with membrane phospholipids, okay, causing changes in permeability and electrophysiologic alterations. Next, let us see about cytoskeletal abnormalities. So cytoskeletal filaments, what they will do is they will serve as anchors. You know, the cytoskeleton helps to maintain the, is nothing but the skeleton of the cell. So they will serve as anchors connecting the plasma membrane to the cell interior, and proteases which are activated by cytosolic calcium will damage these cytoskeletons. So cell swelling, particularly in myocardial cells, will occur, and this damage will lead to detachment of cell membrane from the cytoskeleton and resulting in vulnerability to stretching and rupture. So damage to different cellular membranes have different effects on cells. So mitochondrial membrane damage. See, mitochondrial membranes result in opening of mitochondrial permeability transition pore, lead to decreased ATP generation and release of proteins that will cause apoptotic death. Next, plasma membrane damage. So plasma membrane damage will result in loss of osmotic balance and influx of fluids, as well as loss of cellular contents. Next, injury to lysosomal membrane. They will result in leakage of their enzymes, lysosomal enzymes, into cytoplasm, and it will activate acid hydrolysis in the acidic intracellular pH of the injured cell. And these lysosomal hydrolysis include RNase, DNase, protease, phosphatase, and glucosidase, which will degrade the RNA, DNA, proteins, phosphoproteins, and glycogen respectively, and make the cell move towards necrosis. You will see damage to DNA and generation of free radicals in the next lecture. Thank you.